questions about any of the material from last week? Any issues yet on the homework or anything like that? Okay, then I guess we can move on to this week's material. What we're going to be talking about in this final sort of full module of the first half of the class is the issue of networks and pathways in biology, and what's often referred to as the subject of systems biology. So systems biology is kind of a vaguely defined term. It's something you hear quite a bit if you work in biology today as an organizing principle for how a lot of people are trying to think about biological problems. And although the term is kind of vague, it does capture an important idea, which is that if we want to understand complex systems, and biology is largely a collection of very complicated systems of many components interacting in all sorts of complex patterns, if you want to understand something like that, the idea is that you can't do this just by understanding the components. You can't just look at the individual proteins or the individual genes or interactions between these and understand what 1,000 different proteins and genes and small molecules are collectively doing together. You have to look at this in terms of integrated systems. And so an example of a system might be the cell cycle control system, so a cascade of many genes that are involved in controlling when the cell is in the different phases of the cell cycle, when it divides and replicates, and so forth. Various kinds of developmental programs would be examples of systems we might study in systems biology. So how does a tissue or an organ form? That might be an example of a complex system that would involve numerous different components interacting in a complicated sequence of very precisely choreographed steps. And we're not going to be able to understand how those steps contribute to an overall function if we don't think about this in terms of a unified system. Many responses to different kinds of external stimuli would have similar sorts of behaviors. So what does yeast do if you subject it to osmotic stress? That would be an example of another complex system that we can really understand better by thinking of it as a system rather than as a collection of genes. So this is commonly contrasted with what are called reductionist approaches to biology, which is the more traditional way biology has been done, with individual labs often looking at a single protein or a single kind of interaction and looking in great detail to understand every aspect of how that system works. And the idea is that systems biology is not necessarily going to replace this reductionist biology, but that's something we need if we're going to turn reductionist information into something that actually lets us understand complex systems. One of the reasons I mention this is just because it's a very important trend in biology in general. These systems approaches are really taking over huge fractions of biological research and are becoming an important part of how everyone in biology is doing research today. But I also mention it in this class because we really can't separate systems biology from computational biology. We have systems biology because we have computational biology, and because we had computational methods that made it possible to start thinking about very complicated sets of data, so huge data sets profiling the activities of many genes and proteins and molecules, and actually be able to say something meaningful about what patterns can be drawn from this and how all of this information can contribute to an understanding of the overall function of the system. So systems biology is really something that was created by computational biology and is becoming the standard way people are doing high-profile biological research today. So I think it's an important thing to understand and an important thing to cover in this class now that we've gotten far enough along to understand some of the methods that go into this. Are there any questions about any of that? All right, so as is often the case with computational topics, we need to think about our abstractions. So we're not going to be able to work with complex biological systems unless we make some simplifications that make it possible to start inferring computational or mathematical models. And very often the abstraction we use in talking about systems biology is the network. So networks are basically the same thing that we've referred to previously as graphs. And this is something we've seen in a few different contexts in this class already. Can anyone think of a place we've seen graphs before in this class?
Like, well, So this is not exactly graph, like sort of an HMM, kind of like it.
Yeah, so an HMM is a kind of graph, or involves a graph as a way of specifying how you move between different states of models. That's one place we've seen graphs. Yeah.
Genome assembly?
Yeah, the genome assembly is another example of this. We use graph theory to understand how to build genomes. So working through that abstraction graph and the genome assembly problem gives us a way of bringing in a lot of different computational tools that we can then recognize are useful for solving this hard computational problem. And it's basically the same thing in systems biology. By having this abstraction, even though it is often a simplification of the real system, we get a lot of power for asking questions that would otherwise be difficult to ask, and a lot of tools we can bring to bear on those problems. So a graph, as we've seen before, has a couple of essential elements. A graph has nodes and a graph has edges. In the systems biology context, our nodes might be genes. So we might have our nodes of the graph representing different genes if we're interested in how patterns of regulation function among genes. They could be proteins that would typically come up if we're looking at networks that involve interactions between proteins. In some cases, there's small molecules, such as metabolites or other kinds of products of chemical reactions that would occur in biochemical systems. And that might be something we're looking at if our network is describing networks of reactions in, let's say, metabolism of an organism. Edges, likewise, would describe different sorts of interactions that may be important to us in these contexts. So if we are talking about a network of proteins, then we might be interested in edges that refer to binding interactions between proteins. We might want to know which proteins attach to other proteins, maybe which ones modify other proteins. And then an edge might mean that A is a protein and C is a protein, and this edge is telling us that A has been observed to bind to C. In a gene network, it may be that we are talking about regulation. So it might be that our genes A, B, and C are transcription factors, and we're interested in which transcription factors activate other transcription factors, and those edges might be describing these patterns of regulation. In metabolic networks, often the edges would be describing chemical reactions. So there is a reaction that converts A into either B or C, and then that would be an interpretation of a network in that kind of context. So the same abstraction will often apply to a variety of different kinds of ways of describing interactions between components, different sorts of components and different sorts of interactions. And because of this, we often need different variations on the network abstraction that we're dealing with. So in graphs, there are a lot of different variants on the basic graph model, and one of the ways this is often distinguished is in how we represent the edges in the graph. So some graphs are what are called directed graphs, and directed means that an edge from A to B is different than an edge from B to A. And the alternative would be an undirected graph in which an edge between A and B is the same regardless of whether it's A to B, B to A. We don't distinguish. Graphs can be weighted or unweighted, and a weight would refer to simply a number that we attach to an edge on a graph. And in some cases, we use a kind of degenerate version of weighting known as a signed graph, where we would say we don't care exactly what the value is on an edge, we just care whether it's positive or negative. And all of these would have different interpretations in terms of biological networks we might want to describe. So if we are talking about, let's say, a protein network, so A and B are proteins, and we care about which proteins are binding to other proteins, it might be natural to have an undirected graph. So A binds to B means the same thing as B binds to A, so that would be an example where we would want an undirected graph. On the other hand, for a genetic regulatory network, we might want a directed graph. If A and B are transcription factors, it means something different to say that A regulates the expression level of B than to say that B regulates the expression level of A, so in that case we would need to have a directed graph. In some cases, we will care about weights on our edges, and that too may have different interpretations in different contexts. If we're talking about protein-protein interactions, for example, which proteins bind to one another, a weight attached to an edge might refer to the probability those proteins bind to one another. So often our evidence is ambiguous. We may not be sure whether or not that interaction occurs, and we might want a weight to capture that uncertainty. or it could refer to the strength of the interaction or the fraction of the time they seem to be bound to one another. So again, lots of different interpretations, but often these can be fit into these models of basically a graph or network. There is sometimes specialized notation in these graphs that's sometimes used in biology, so I'll just point that out. So when we are talking about signed networks, sometimes you will see an arrow that looks like this, or a kind of bar that looks like this. When we're talking about signed biological networks, this particular notation means a negatively signed edge, and this one would mean a positively signed edge. So in a regulation sense, what that would typically mean is that we're referring to upregulation or downregulation. So an arrow from A to B would mean that A upregulates B. So if you have more A, the level of expression tends to go up, and the bar would mean A downregulates B. So more A causes less B. So is this clear to everyone? All right, so there are a lot of reasons why we would want to use this kind of abstraction, even though it is often a simplification of the real system. One of the reasons is that there are a lot of kinds of analysis methods that are broadly useful for looking at many kinds of networks, and by recognizing that we can represent our problems as networks, we can bring these tools to looking at diverse kinds of biological networks. So an example of this that got a lot of scientific coverage a few years back is the idea of network motifs, that we can look at lots of different kinds of networks and recognize some common sorts of simple patterns in those networks that tell us something about functions of those elements. So this is an example of a motif called a feed-forward network, where we have a particular pattern among 3 nodes of our network of upregulation and downregulation that turns out to be very useful for producing finely controlled responses to external stimuli. And this kind of pattern is found to be statistically overrepresented in broad classes of networks, including genetic regulatory networks, protein interaction networks, neural networks, as well as various kinds of non-biological networks, such as communication networks, social networks, and so forth. So that's one reason we look at this. We can recognize that some of the analysis tools built up for networks in general give us insight into biological networks specifically. There's also a lot of theory on statistics and algorithms for graphs or networks. So there are various kinds of statistical properties of a graph that people who have worked for many years on graphs can recognize are often useful ways of capturing properties, such as how easy it is to communicate across distant points in a graph, or to recognize how robust a graph is to damage or perturbations. And by recognizing these kinds of things, we can get an idea of what properties biological networks have evolved to have. Likewise, there's a lot of computer science theory on graph algorithms. We saw an example of that in the sequence assembly context, where we could recognize that by converting our problem into a problem in graph theory, we can then bring in all this algorithm machinery that was developed for studying completely different real-world problems, but using similar kinds of abstractions. So again, this is something I can't really go into great depth about all of the kinds of algorithms here. If you take any sort of algorithms class, I can almost guarantee you'll be seeing a lot of graph theory there. Any questions at this point? There are also a lot of tools for learning network models, and again, by representing things in this way, we can bring in some of this kind of theory for how you learn a network to these biological domains. In the next class, I'll be going through how some of that theory works, talking specifically about the problem of how we can learn networks that are involved in genetic regulation. So you'll get to see some of that, but basically there are these different classes of models, such as what we call Bayesian graphical models, that provide useful ways of using a graph to organize interactions among large sets of components, and that give us a lot of computational machinery for working with those models. There are similar things for learning the weights of the models using a technique called regression, and a lot of kind of statistical and machine learning methods that have been developed to learn these kinds of graph problems, to figure out which things are interacting with which, how strongly they're interacting, how confident we can be in their interactions, from all sorts of complicated, noisy data sets. And by recognizing this, we can bring all of that to these biological domains. finally, there are some common kinds of properties that have been observed in many different networks, and by recognizing these properties and knowing to look for them, we can get insights into biological networks. So many networks, for example, have been observed to have a property called scale-freeness, or being a small world network. This refers to a particular kind of pattern you often see in networks that has to do with the degrees of different nodes, basically how many things are connected to any given node in a network. And networks that have this property tend to be very good at propagating signals efficiently across many different components of the network. And by knowing that this is an interesting property about networks, we can know to look for it in biological networks, and often we find that biological networks have this kind of property. So all of that is basically just motivating why we care about networks. And what I wanted to spend most of the time in this class on is talking about some of the kinds of networks we'll often be seeing in biological contexts, as well as some of the biotechnology that we need to work with if we want to use those computationally. So there are any general questions about networks before I move on to that? Okay, so one of the kinds of biological networks we'll see quite a bit, which I've already mentioned a few times this class, is a genetic regulatory network. And a genetic regulatory network is simply a network that describes patterns of regulation among genes, very typically transcription factors. So we've seen previously at a coarser level of detail some of the features that go into, in this case, eukaryotic transcription, so basically eukaryotic promoters. And a promoter, as we saw previously, will typically have a set of sequence signals that will help the cell identify that this is the site of a gene, so it's something that should be transcribed. And some of these are in the form of what are called, generic binding sites, so things that are recognized by what are referred to as general transcription factors that you more or less find in every gene. They're not 100% conserved, but by and large, usually a eukaryotic gene will have these basic kinds of signals, and there will be some standard transcription factors that are always present that recognize those signals and help the cell figure out that it's looking at a gene. But then there are other specific transcription factors that recognize sites that may or may not be found in any individual gene. And these are commonly organized into what are called silencer and enhancer sites. So silencer site would be something that's typically used to turn down activity of a gene, and enhancer is a site that's typically used to turn up activity of the gene. And these are recognized by what are called specific transcription factors. Repressors that recognize silencer sites, and by binding to the silencer turns down activity of the genes. Activators that recognize enhancer sites, and by binding to the enhancer turn up activity of the genes. So that is basically the machinery by which cells control transcriptional regulation, and this sort of pattern of regulation gives rise to one kind of network we're often interested in, which is essentially the kind of network that describes how patterns of gene expression across the genome respond to cascades of signals in terms of expression of changes among these transcription factors that can affect other transcription factors and affect various downstream genes. The actual system is quite a bit more complicated than that abstraction. So we have repressors and activators, but these repressors and activators themselves may act differently in different sorts of contexts. A particular protein might be a repressor for some genes and an activator for other genes. It may be a repressor some of the time for one gene and an activator some of the time for the same gene. These are also influenced by co-repressors and co-activators, which are other proteins that interact with the repressors and activators and may modulate their behavior, either making them function. These often act combinatorially in a single gene, which would mean that in any one gene, we might have multiple different transcription factor binding sites that respond to different transcription factors and different levels of these transcription factors. So you might have a single gene that has multiple sites for, let's say, the Kreb transcription factor and a binding site for the ZIF268 transcription factor. and the interactions among these different transcription factors and the ways they can interact with one another or block one another from getting to their binding sites or assist each other getting to their binding sites can lead to quite complicated regulatory logic. In a network, we often abstract away these details. We say either there's an interaction or there isn't, has a certain strength or it doesn't. But these can be implementing complicated kinds of behaviors where it may be that you need a particular narrow threshold of Kreb expression level and a particular threshold of Zip expression level to get expression here, and it may be much more finely modulated. It's often not really just on or off. You can have fine levels of modulation of the gene that's being regulated based on the levels of the genes that are regulating it. Any questions at this point? All right, so to see how some of this works, I wanted to walk through one example of a pretty simple genetic regulatory network, but one that I think illustrates how even the simple things can get pretty complicated when you start looking at them more systematically. So this is a network many of you may have seen in one context or another earlier in your studies. So this is the phage lambda repressor network. And a phage lambda is simply a bacteriophage, so it's a virus that infects bacteria. And when lambda infects a bacterium, it injects its genome. And what happens at that point is that the bacterium goes into what's called lytic cycle, which basically means that the genome kind of sits dormant in the bacterium, and it's just sort of waiting for some kind of signal. So this may last for quite a while with the genome just sitting there with no actual virus present aside from genetic material, but eventually something changes and flips this into what's called lysogeny, where the genome starts transcribing the pieces to construct new viruses. You start getting new phages growing within the bacterium, and eventually you get lysis when you get enough of these to burst the bacterium, and you get new phages that go on to presumably infect other bacteria. So that is basically the lysis-lysogeny cycle you're likely to have seen or likely are going to see in other classes. But I'm interested in this for today because it gives a nice, relatively simple example of what a regulatory network might look like. So this is an illustration of the regulatory network involved in controlling this switch between lysis and lysogeny for phage lambda. And basically, there are just two proteins involved in controlling that switch. There is CI, known as the lambda repressor, and CRO. And these are both effectively transcription factors that act on the genome of the virus. And what they do is as follows. So the lambda repressor is an activator for itself. So lambda repressor binds to a binding site that is upstream actually of both of these genes, the lambda repressor and the CRO gene. And when it's bound there, it promotes expression of its own gene, and it represses expression of the CRO gene. And CRO also happens to be in a common operon with various other genes that are involved in producing lysogeny, so growing the new phages. So it is also repressing these downstream lysogenic genes. CRO has the opposite effect. So CRO represses lambda repressor and activates itself as well as these downstream genes. So that basically is the regulatory behavior that's involved in controlling the lysis-lysogeny switch, and that's an example of something we can understand through a simple network model. So just by a show of hands, how many of you have seen this before? Okay, so about half of you. So this is one of the canonical examples of how this kind of switch functions in biological systems, but I want to look at it now in terms of a network. So this would be an example of a network that would describe what I was just telling you about how this switch works. So this is simplifying quite a bit. It's throwing out a lot of details that exist in the real system, but it's describing how this behaves. So can anyone tell me, is this a directed or an undirected network? So this is a directed network. So it means something different to say that lambda repressor represses CRO than to say that CRO represses lambda repressor. It's also a signed network. I've added these pluses and minuses to kind of illustrate that more freely or more clearly, but we also have these positive edges represented by arrows and the negative edges represented by bars. So this is saying we have two proteins here, Lambda repressor and Cro. Lambda repressor, it upregulates itself and downregulates Cro. Cro upregulates itself and downregulates Lambda repressor, and Cro upregulates these various proteins that are involved in producing the switch to lysogeny. So let's say we have the system in a particular state. So we're going to assume the bacterium is infected, it's in the lytic cycle, so we've got a lot of Lambda repressor and very little Cro. And we can look at this network and it would tell us something about what happens when we're in that situation. We have a lot of Lambda repressor, that is upregulating itself, so it's kind of locking itself in. Having lots of Lambda repressor creates more Lambda repressor, and it prevents the genome from transcribing Cro. So you get the Cro kind of locked out. You have very little Cro present. Because of that, we don't get repression of Lambda repressor by Cro, and we don't get promotion of these lysogenic proteins. So this is kind of locked in that position of the lytic cycle. But let's suppose something comes along that behaves as follows. So some protein comes along and represses Lambda repressor. Based on this network, can anyone tell me what you would expect to happen then? Yeah, so if we had something that came along and repressed the Lambda repressor, the Lambda repressor goes down, you get a kind of positive feedback there because the lower Lambda repressor goes, the less able it is to drive its own expression, so it gets lower and lower. That reduces this inhibition of Cro, so Cro starts coming up, and there you also get a positive feedback. Once it starts coming up, it can start promoting even more expression of itself, so it gets larger and larger. Cro represses the Lambda repressor, so you basically flip the switch from Lambda repressor to having Cro. The Cro comes up, it promotes itself, it blocks the Lambda repressor, and it starts the production of the lysogenic proteins, so the phage can start producing more phages and eventually get you to lysis. So that's kind of how having a network model can help us reason about how some new change or some hypothetical interaction in the system might affect the overall behavior. And often we would be interested in these models for that kind of application. So to consider what might happen if there was some kind of change or a mutation that knocked out one of these edges, or if you did an experiment that, let's say, put in something that would reduce or increase expression of one element of the network. Networks help us reason about what might happen in those cases. And when we get into more quantitative models of the network, you can say that in much more detail by really simulating behavior of a network at a quantitative level and making predictions that maybe we couldn't make just by looking at the network. So any questions about any of that? All right, so these things can be quite a bit more complicated than that example. You often have multiple layers of regulation. So a quite typical sort of way these networks might work is that there is some early stimulation, let's say an external stimulus like a hormone molecule docking to your cell, and that activates some what are called early transcription factors. They're kind of the initial response. They will activate later transcription factors that themselves turn on different modules of genes that are necessary for producing whatever response that hormone is supposed to elicit within the cell. Examples of this are available for many kinds of systems. This is an illustration from one of the early systems biology studies trying to put together these big networks. In this case, putting together the cell cycle network for yeast. So this was a network that involved about 800 different genes. And this is not the standard way we would typically visualize a graph or a network, but this is basically an alternative way of viewing it, where you can kind of think of different points left to right here as representing different genes. And then we have arrows showing how genes active at different stages in the cell cycle reach across stages to turn off the current stage, turn on the next one, and sometimes reach farther along to kind of get things ready for subsequent stages of the cell cycle. So this is more of an illustration of how complicated these things can actually be. And of course, for something like a mammalian cell cycle, even this would be a simplification of the sorts of things we might be seeing in these very complex cases. And so any Questions about genetic regulatory networks? All right, so one last thing I want to mention about these is how we learn these. So we'll be talking next time, as I mentioned, about some of the computation that goes into learning them. But for the computation, of course, you need data. And this data can come from various directions. A lot of our data is actually technologies we saw last week. So in many ways, how we go about learning these networks is by looking at patterns of expression. And usually we're going to need some high throughput expression assay. So microarrays or RNA sequencing. And then we use patterns of correlation, basically looking at how expression patterns of different genes change concordantly or non-concordantly under different conditions or different points in time to figure out which genes are under common regulation, potentially what's regulating them. Often we need to combine this with other kinds of data that tell us more specifically which transcription factors are binding to the promoters of which genes. And often that comes in the form of a direct binding assay. And these commonly work with a technique called chromatin immunoprecipitation, or CHIP, and this more or less is done in two different variants. What's called CHIP-CHIP, which means using chromatin immunoprecipitation with a microarray, or CHIP-seq, which is using it with a sequencing study. And in either case, basically the way it works is as follows. We have our genome, and we would assume transcription factors are binding to the promoters of the genes they regulate. So if you have your genome, you would expect you'll have various transcription factors bound to different points in the genome. You can use a chemical modification then to fix your transcription factors to the genome. So add a chemical such as formaldehyde that forms cross-links between different molecules, so everything in the cell kind of gets stuck to whatever it's attached to. So your transcription factors get stuck to the genome wherever they happen to be sitting. And you can then fragment your genome and immunoprecipitate. So use an antibody to your transcription factor to pull out all the pieces of genome that have the transcription factor of interest stuck to them. And then you wash off your transcription factors and you just ask, what are these pieces of sequence? You can do that by running the fragments that you fished out through a sequencing machine and blasting them against your genome, or you can do that by running them over a microarray where you have spots that would be identify promoters of different genes. In either case, what you get out is, in theory, a measure of what places any given transcription factor is binding in the genome. So that's a very useful kind of data to have if you're trying to put together a genetic regulatory network. One other source of information is actually other kinds of computational prediction. So these transcription factor binding sites tend to have conserved patterns to them. They're recognized, they're sites on a genome that are recognized by a protein, and they will have some regularity to the pieces of DNA they recognize. They may need some particular sites with very high conservation. Some sites may be quite flexible, but more or less the transcription factor is sitting on a region of genome and recognizing a pattern there. And computational methods tend to be very good at identifying those sorts of patterns and taking examples and generalizing to figure out what other DNA strands that same transcription factor would recognize. So a lot of what we would typically do in putting together these networks would involve building these kinds of computational predictions, and this can be done using techniques we've already seen in other contexts in this class. So you may remember position-specific scoring matrices that we saw in the context of trying to identify protein domains. So these are a technique for identifying this sort of pattern in a sequence, and this works quite well for identifying patterns in DNA sequences too, and is often used as a way of identifying transcription factor binding sites. So typically when we're trying to put these together, we might be combining multiple sources of information. We might have some RNA-seq data, some binding data, and some predicted binding sites. We would want to bring all of that together to try to put together a genetic regulatory network. And again, we'll see more of the computation behind that next time. Any questions about any of that? Another kind of network we'll often be interested in is a protein-protein interaction, or PPI network. So a PPI network would have nodes that represent proteins and edges that would typically refer to binding or some other kind of interaction, such as a modification a protein produces on another protein. In some cases, these PPI networks describe formation of complexes of proteins. So if we have, let's say, a set of proteins, such as these three here, that bind to one another to form various protein dimers, so pairs of proteins, that's a kind of interaction we can describe by a network. We can say that we have a network where our nodes are the proteins and our edges are the interactions, so all of the ways that one protein in our node set can bind to another protein, and then that network would describe, to some degree, the way these different proteins can participate in forming complexes. As is often the case, the network is removing some details that may be quite important to us. So if you looked at this network, there would be no way of knowing that these three proteins form dimers, so pairs with one another, and don't form trimers, that you never get three of them forming into a complex. So that is another way that the abstraction does lose information, but it's still often a useful abstraction to have. One particular kind of network we often see in the protein-protein context is what's called a signaling network. And this is a network where interactions very often take the specific form of phosphorylation and dephosphorylation. So many proteins are what are called kinases, proteins that bind to other proteins and attach these phosphate groups to the proteins. And other proteins are phosphorylases that remove phosphate groups from proteins. And this process of attaching and removing phosphate groups is one of the major ways that protein activity is regulated, and this often contributes to what are called signaling cascades, by which some external stimulus, or in some cases internal stimulus, can be transmitted from different parts of a cell to one another. So you may have what's called a G-protein coupled receptor, a protein that sits in the surface of a cell and recognizes an external signal, like a hormone docking with that cell, and it will be a kinase that binds to another protein and will produce, say, one of these kinase reactions where another protein becomes phosphorylated. That phosphorylation changes the behavior of this second protein, known as a relay protein, and causes it to start phosphorylating other proteins. So the relay protein is both the target of a kinase and itself a kinase, and it may activate another protein called a messenger protein that itself is a kinase that moves to another part of the cell when it's phosphorylated and phosphorylates yet another protein, which amplifies a signal, and eventually gets down to phosphorylating the things that act later in the network. So these may be the actual functional proteins that need to respond to the signal. They might be transcription factors that then turn on other kinds of proteins through genetic regulation. But that basically is a signaling network, and that's often something we're looking at specifically when we talk about PPI networks. Any questions about any of this? So this is an example of some pieces of PPI specifically related to cancer signaling. So these kinds of signaling interactions are often disrupted in cancers. This is from a database called the KEGG Pathway Database. We'll be looking at this one, among others, on Friday. And this is illustrating some of the ways that an external cellular signal, in this case, peptide molecules that are secreted by other cells, can interact with cell surface receptors, so that's these green boxes here, and trigger these cascades of signals and other interactions that eventually converge on functions such as increased proliferation of the cells. In the cancer context, we care quite a bit about this because these networks are often disrupted in various ways. You get aberrant signaling, such as, for example, something going wrong with these proteins that causes them to keep triggering the proliferation signal, even though they shouldn't be. There are lots of other contexts where we care about this. So this is just an example of what a PPI network might look like. These traditionally have often been learned from a kind of assay called a yeast-2 hybrid assay, at least when we were learning them on large scales. This is a kind of assay that gives us a way of figuring out if two whose presence we can detect, breaking it in half, and then attaching the two halves to two proteins where we want to test for interactions. If those proteins bind to one another, they bring together the two halves of this specialized protein, which is a transcription factor that activates a signal we can detect in the cell. And so basically, you can do this with lots of variants of the two proteins we're trying to test, see which pairs give us a signal, and then based on that, figure out which proteins are likely to be binding to one another. That gave us a way of looking at relatively large scales for this kind of interaction, but it's also a very noisy method. There's a lot of uncertainty, lots of false positives, so it says things bind when they really don't, lots of false negatives, so things that really bind for one reason or another don't produce the signal at a detectable level. But this was kind of the first thing that really allowed us to start putting together these PPIs on a large scale. A lot of what we know about them is done more manually by the kind of reductionist, more hands-on studies. So often you would figure this kind of thing out by what's called co-immunoprecipitation, to basically use antibodies to sort of fish out pairs of proteins, and then see if you're more or less fishing out one protein will bring out another protein that is of interest to you, and hint that they may be bound to one another. So lots of ways this kind of thing can be used to build your network more slowly, and often we would really need this kind of technique to verify our network because there's so much uncertainty in the high throughput technologies for this kind of study. There are other ways of doing this, but these are a couple of examples. The third major kind of network I wanted to mention is metabolic or reaction networks. A reaction network would describe a network in which our nodes are typically small molecules and our edges correspond to reactions, typically reactions transforming one molecule into another. So here we might have a system where we've got molecules, A, B, C, D, E, F, and G, and various reactions such as A plus B goes to C, and we can produce a network that describes how these reactions behave. Just like with the PPI and the genetic regulatory case, the network isn't capturing everything, and you would often have to have other information beyond the network to really know what's going on. But we might have, for example, an interaction such as A plus B goes to C, described by an edge saying A is transformed into C, and an edge saying B is transformed into C. And we wouldn't necessarily know there if C is built entirely from either A or B, or if C is built from both A and B. We'd need other information that's not caught in the network abstraction to do this, but still, it gives us a good way of making sense of very complicated kinds of networks of interactions. And this sort of thing is often used in contexts where we're trying to simulate complicated biochemical networks. These can also have various kinds of regulation within them that might influence the networks in different ways. So in some cases, in addition to just the basic reactions in the graph, we might be interested in ways that, for example, one component of the network might regulate production of other components. So it's quite common in, let's say, a metabolic network that something produced at one step in a pathway will regulate the production of something earlier in the pathway. For example, we can have allostery or competitive inhibition, by which this molecule E might shut down production of molecule C when you have too much E. So that also is something you will often see in these networks, these kind of regulatory edges in addition to the direct reaction edges. Any questions about that? Okay, so this would be an example of a metabolic network. So this metabolic network is describing a set of small molecules here. I know most of you probably can't read the labels of these molecules, but we have sets of molecules like fumarate, acetalate, oxaloacetate, and so forth, with edges describing reactions, which would be annotated typically by the enzymes that promote these reactions. And then we have interactions among all of these different kinds of things in these complicated sorts of cascades of biochemical reactions. For any of you who have taken biochemistry already, does anyone recognize what network this is?
The Kreb Cycle?
Yes, this is the Kreb Cycle, and all of these kind of go together. This is also the citrate or TCA cycle. Basically, all of these things kind of integrate together. And when we get to the databases, we'll see that basically you can get the entirety, or pretty much the entirety, of the conserved pathways of metabolic networks, all linked into these huge networks that you can explore. And we'll see how to do that on Friday. So these are not quite as easy to learn. We don't really have good high throughput methods the way we do for the genetic regulatory networks, and to a lesser degree the Ppi networks. Things are kind of getting there with mass spectrometry, so you can profile expression of large number of metabolites over time using mass spec, and that gives us ways to bring in some of the same kinds of computational methods, but most of what we know of these has been worked out more manually by kind of hands-on biochemistry. Those are your classic studies of one reactant or one enzyme at a time. But computational methods are being used quite a bit today in understanding these, in learning, in some cases, the structure of the network, so what reactions might be producing what things, and often learning rate constants. So what are the rates of interactions, what are the different probabilities of different pathways. Often this is tightly integrated with simulation methods, something you'll learn about in the second half of this class. And there are also specialized methods such as flux balance analysis, which is a technique for using the structure of the network to kind of constrain the possible, basically the possibilities of what might be happening and make it possible to infer complex networks from relatively small amounts of data. The last thing I wanted to talk about is some new directions in these things. So those three kinds of networks are really the main kinds we will see in biological systems, but there are a lot of other kinds of things that are described by networks, and some of them we're realizing are quite important. So I wanted to kind of quickly go through a few of these emerging directions in networks and pathways. One of the big ones refers to regulatory RNAs. So transcription, we know, is often regulated. You will often have activities of genes regulated at the level of transcription by transcription factors. But expression is regulated in other ways as well, and it's become apparent that small RNAs are very important for regulation of gene activity at a different level that has to do with lifetimes of transcripts in the cell. So basically regulating how quickly particular transcripts are degraded within the cell. There are different versions of this, different examples you'll sometimes see, but a canonical one that we've realized is quite important in humans as well as other eukaryotes is something called a microRNA. And the way a microRNA works is that we have RNA genes, so genes that just code for pieces of RNA. Sometimes these are kind of hidden in introns of other genes, but these are chopped up by something called a dicer, which produces little fragments of RNA, double-stranded RNA, that serve as templates for something called a RISC complex that basically tells it what genes it should be regulating. So the RISC complex will take up one of these little fragments, and it's like an instruction that tells it what it should be trying to degrade. The RISC complex will use that to recognize binding sites in transcripts that are floating around in the cell. Typically, but not always, these are in the 3' UTR, and when it recognizes the thing it's looking for, it will degrade the transcript and in that way turn down its expression. This was something people didn't really appreciate was important until we had a human genome and could start to see how common these things were in the genome. But now we understand it's a very important part of how our expression of the genes is regulated. There's a lot of interest in integrating this into our other kinds of regulation. Splicing regulation is another emerging area, so another way that we've started to realize that in addition to the kind of standard generic splicing signals, there are a lot of specialized splicing signals that allow the activity of splicing to be controlled on a cell type specific or condition specific basis. So what are called splicing silencers and enhancers that may be exonic or intronic, and that respond to different signals to allow a cell to shift between different splice forms of a gene in response to various signals or cell cycling or developmental stages and so forth. So this is another kind of area that's becoming important, or it's always been important. We're realizing how important it is. Chromatin remodeling is another area of regulation. So our genomes are normally not freely accessible in the nucleus of a cell. They spend most of their time tightly wrapped around protein complexes called histones to form something called nucleosomes. And actually a key step in expression of a gene is unwinding these histones to expose the regions of DNA. And we're finding that this too is a very important component of how gene expression is regulated on large scales within the genome, and that too is another emerging area of study. understanding these patterns of histone regulation, how they're controlled and how they differ under different conditions. RNA editing is another one of these emerging areas, a process by which transcripts, after they're produced, can actually have their sequence altered by specialized proteins. Sometimes this is constitutive, so a particular transcript is always altered in a particular way, but often it's regulated. And this is really an emerging area where we don't know a lot about the regulation of this, but we know it exists. It's a way that cells can change their behavior, and often this is important in some contexts like development or behavior neuroscience particularly. So a lot of what's going on in the field today really isn't about looking at one kind of network, it's about integrating all of these different kinds of networks, which are often tightly intertwined with one another, interacting with one another, and we really need to bring all of these directions, both the classic and the emerging ones, together if we want a real understanding of our biological system. The kinds of computational methods we use are often very well suited for working with these heterogeneous data sets, and often actually work much better if you have different lines of evidence and different kinds of networks you can bring together that all sort of support a common picture of how a complex biological network works. So I know we're out of time at this point, so I'll just leave you with that thought, and then next time we'll see some of the ideas that go into how we actually assemble these networks. Any questions? Okay, and I'll see you on Wednesday.